speakers. You just heard that there will be a recording of this session. Would you have at least would you not be allowing us to publish this afterwards on our channel? Please tell us. So, as I said, we will have two speakers. The first one will be Dr. Ashenafi Gunnabo from Ethiopia, and his topic will be the adoption of Rhizobium inoculant technology in southern Ethiopia. Ashenafi is an assistant professor of plant and microbe interactions at Arba Minch University in Ethiopia. He's earned a PhD in microbiology with a focus on plant-microbe interaction from Wageningen University in the Netherlands, and his master was from Hawassa University in Ethiopia. Our second speaker, Sofie Goormachtig, will be talking about capturing rhizobia to introduce high protein yielding soybeans in Europe. She's professor at Ghent University and a principal investigator at the VIB Center for Plant System Biology. Her research investigates the beneficial interactions between plant roots and microorganisms, with a particular focus on improving plant growth under stressful conditions. She's also involved in the development of soybeans as new crop for Northwestern Europe, and she's a co-founder of the biotech companies Afea Bio and Protealis, which aim to apply cutting-edge microbial technologies to agriculture. So we will now have the two speakers, one after the other, and afterwards we will have an open discussion talking about these two presentations. You can post your questions already during the talks in the chat. We will make sure that we pick them up afterwards. And if not, we can still have, we can open the discussion with those questions and have more questions afterwards during the discussion. Without waiting longer, I would like Dr. Ashenafi to please share his presentation. Onesimus, you will need to unshare. Thank you very much.
Okay, thank you very much for inviting me for this nice presentation to share what we did in Africa and Ethiopia. And I would like to introduce what we are doing really in Ethiopia. So I want to share the slides. Yeah, I have. Okay. Okay, you see my slides?
Yes, we do see your slides. Thank you. You're still not in presentation mode though?
Okay, I will make them. So can you see now?
Yes, perfect. Thank you.
Okay, let me check that. It's moving, right?
Yes, it's working.
Yeah, okay. So I will be handling the African case, and I would just like to introduce myself. My name is Ashenafi Hailu, as introduced. I was born in southern Ethiopia, specifically in Gidole, which is some 50 kilometers south of Arba Minch, the regional capital of southern region. And I started primary school there in Gidole and went to Arba Minch for high school. I did my BSc and master's degree at Hawassa University, and I moved to Wageningen for PhD. Okay, so I worked at Southern Agricultural Research Institute for 3 years as a research assistant on aquatic culture aquaculture, and feed for fish. And then I went to Walaita Sodo University and worked as an assistant lecturer there, teaching applied microbiology courses. And then I moved to Arbam University since October 2020, and I'm working as an assistant professor of plant microbe interactions there at Hawassa Amnat Arbam University, which is very scenery place where many people would like to visit, and I would like you have some time to visit this area. Well, my work at Arbaminchi University is basically supported by Arbaminchi University and Fund for Innovative Development from France. And with this, I will briefly introduce what we are working. First, let me briefly highlight to you what legumes are in Africa. Basically, they are produced as subsistence food for farmers, and they are primarily meant for food. When they are actually produced in surplus, they went to the market, and they can constitute as the nutrition and food insecurity for the communities. They are also being used as the forage for livestock, and also a good source of nitrogen fixers, since they supply fixed nitrogen to the soil, and they are used in soil fertility. In Africa, the research basically focuses on grain legumes, which are also being inoculated by various rhizobia, but they are not actually limited to legumes. They are also including, the grain legumes, they are also including the forage legumes, the tree legumes, but they are rarely inoculated with rhizobium. So much research focuses on studying the rhizobia nitrogen fixation potential and their contribution to soil fertility. But the work has not been really developed into inoculant technology and established in African countries. And this is one of the lacking facilities that we need to work on. So some studies really reveal that microbial inoculants are encouraging in farmers' field, and they are promoting the crop yield in the farmers' field. They were very successful in some locations, but in some areas, they were not as such effective. And local isolators, for example, from East Kenya, West Africa, and Southern Africa have been reporting local inoculants have not been working well compared to other commercial inoculants, which are imported actually from other areas. So it was interesting to see while some areas are effectively working with rhizobia, but other locations are not that much performing well. And they have put reasons for this. One of the reasons is indigenous rhizobia are either ineffective, or they are competitive, or there are poor shelf life of the inoculants that have been applied in different areas, or farmers have no awareness to use those inoculant technology. And there is also a huge gap between researchers, extension workers, and farmers regarding the developed inoculant usage across Africa. And this really needs an intervention. And there is also a less developed distribution network between researchers, private partners, African standard organizations providing regulatory frameworks, and there is also a lack of human capacities. All the research work that has been done in Africa is actually supported by the PhD and MSc students working for different organizations. And after completing the project, those works are being limited. So I scan across some inoculants developed in Africa. And it's interesting that South Africa is leading in Africa by having a number of companies involved in inoculant development and production. However, in Ethiopia, we have only 2 companies, which is called MBI and Biosafe, which are recently developed, and they don't have sufficient capacity to reach all Ethiopia, because Ethiopia is a populous country of 120 million, and 80% of the population are dependent on agriculture, which is subsistent. But the government needs to transform this agriculture and it's included as a strategy to inoculate, develop inoculant technologies such as biological nitrogen fixation. But these resources are limited to the center where facilities are, and the areas far away from the center have no access. So my interest was to adopt the Rhizobium inoculant technology to southern Ethiopia and to work in farmers' field. And we planned to renovate laboratory at Arnhem University, because we have nothing there and there is no inoculant use so far. And we want to establish greenhouse, which was not also there. And we want to survey the legume production potential, the usage of inoculant, and also explore rhizobia to screen illets for inoculant development. And we have also planned to train farmers, demonstrate inoculant performance on the farmers' field, and also network, create partners with different organizations like the one we met with the Ghent University, with Sofie. So we are now working and expanding our activities. So my goal at Arnhem University is to develop or establish a rhizobiology center and improve farmers' productivity from there. So as my priority objective, we renovate laboratory, which is now capable. This lab was initially a lab designed for undergraduate studies. Now we renovated it for microbiology lab and molecular biology lab. So you can see we have been trying to develop and facilitate our work. We also established the tissue culture lab and the greenhouse facilities that can support our work at Arnhem University. So then establishing the greenhouse facilities, the lab conditions, and then we started survey of the legume production potentials in the region. One of the reasons we did was that the government have a national plan that is PELS strategy from 2019 to 2024, a 10-year development strategic plan, a pathway to prosperity 2021 to 23, and the agricultural grant transformation plan second, which includes, all of them includes different legume species, including faba bean, field pea, lentil, soybean, groundnut, and a lot others. And they also planned to be competitive in exporting high quality legume products, but with the introduction of using biological nitrogen fixation. So these are some of the reasons why we started this project. And the other reason is that innovative technologies are limited. As I mentioned earlier, the quality of inoculants are poor and no quality check at all in the country. And we dream to have a center. From this center, we can also establish a quality check center. And the remote areas that I'm living in, like around Arnhem, inoculants are not being there. So the survey is actually, the availability of inoculant is limited there, no access to the inoculant in the south, and poor information available on the use and potential of the legumes. So we want to know the potential, uses, and gaps exist to build up and make a kind of intervention to promote the legume production systems. So we had a strategy. One is to design a structured survey questionnaire, and we also defined sample sizes to collect, which was about 847 farmers. We did a purposive sampling based on legume production sites, and we integrate the questionnaire into the Kobo tool to collect the data. We trained experts from agricultural extension centers. We visited to different sites. We contacted extension workers, agricultural sectors, and site selection, farmer selections were performed along with the agricultural experts. To give you a brief account of our respondents, we had a number of household heads which are male dominated, because in Ethiopia, households are dominated by male, and 93%, nearly 94% of the households are male, only 6% are female. And the age category varies from 21 to 83% for 83 for the male households, and for the female households, the household head was 30 to 55 M. Educational status of those respondents were quite varying, but majority of them are illiterate and primary school attended households. Others, they were very small. And then we assessed the farm size that the households have, and most of the farmholders have less than 2 hectares. It's only the farmers who are living in North Ari in the southern Omo Valley, they have greater than 2 hectares per farm. And we try to see the crop production based on the land cropped. And we did a calculation based on the land they owned, but we have also assessed information on how much land was cultivated. So we calculated the crop production, and as land increases, the production of the cereal crop is increased. But when the land increases, the production of legume crop is decreased. This really matches with the concept that legumes are mainly produced for home consumption. So if they are promoting for market, it would have increased with increasing farm productivity. And we have also seen the potential of farmers in growing different legume species, and common bean was one of the most important. If you see the figure A, it was about the total legume production in the area, coverage across all entire region. But if you see B, it is Gamo zone, and if C is one of the zone which is called Conso zone. And in all cases, common bean, here we call it bush bean, and bush bean are also the much common crops that are grown there. And other legumes are grown, but in few concentrations or in least potentially, such as cowpea, groundnut, and mango bean. We have also tried assessing the potential of the legumes and other crops as a priority crops for the householders. And for all, maize was one of the most important priority crop, followed by sorghum, wheat, and barley, teff. The second priority crop was common bean, one of the most important legume crop grown across the region, but it has never been inoculated, never been supplied with chemical fertilizers. And still the third priority crop was also common bean, followed by teff, and others have no third priority crop in the region. And we also try to assess the input use by different locations and regions. So we were able to understand that all farmers were able to use less than 50 kilograms per hectare in their farm. And this is because of the fragmentation of the farm that they are using. And these have no return after investment, is very poor because they are only consuming few crops. There are only few farmers who use greater than 100 kilograms per hectare for fertilizer. And few farmers are also using fertilizers and pesticides. Regarding the Rhizobia inoculant usage, it's only 3% of farmers heard about the inoculants, but they never used. Surprisingly, we find agricultural offices, they have packages of inoculants, but they never tried in their fields. So this is one of the gaps that we wanted to study this work. We were also able to identify the months that the community faces food shortage. And it is actually from end of January to the end of June, farmers face food shortage in the region. And this was because of the rainy seasons. In this period, it was a rainy season, and farmers usually move to the field. But in the drier season, farmers usually have less food so that they can depend on what they produce during the drier season. And we have two spells of rainy falls, one in April, starting from March to July, to June, and the other starts in September. So this is one of the reasons for looking into the farmers. And we also identified that problems in the farm, pest, weed, disease, drought, and other problems are mentioned in the farm as one of the severe cases that need intervention. So after identifying those conditions, we then started exploring Rhizobia from different locations. We traveled to 22 different districts. From each district, we identified 10 farmers and 5 to 10 nodules were collected from fields. We collected from field nodules. We also brought samples and trapped Rhizobia from the soil using plant trap method in the greenhouse. And then we used handbook for Rhizobia and working with Rhizobia manuals to extract Rhizobia from the nodules that we brought from the fields. And we preserved it. Over 1,000 Rhizobia strains were isolated from different locations. We preserved it in our greenhouse, and we also authenticated them using the Leonard jar, which we have modified and adopted according to our local conditions. And based on that authentication test, we have able to include those non-nodulating negative control which have not been inoculated. The positive control is supplied with potassium nitrate, and the one our test was the inoculant, which forms nodules and legumes. And we collected those nodules, counted, and estimated their biomass. So we did some phenotypic characterization, physiological, biochemical entry tests, and metabolism of different strains were conducted. And we analyzed it using numerical taxonomy, and we were able to do the analysis for all tested legumes. For example, for common bean, we have 3 major clusters based on phenotypic properties. One of the most important strain from this cluster was AMU23, which is within cluster 3, which have distinct properties, and it performs well under field conditions. For chickpea, we have 5 different clusters phenotypically, most of them clusters with Rhizobium shonensi, which was isolated actually from 3 legume at Hawassa, but most of our work is related to this species. And within this cluster, AMU73 was able to form an effective symbiont, and we selected it as an efficient strain to work in the field. We did the same analysis for lupine, for tree lucerne, for vetch, and for alfalfa. We have also other legumes that we have included and analyzed their effectiveness. We also tried to see if they are affected and varying based on effectiveness on morphological parameters and also field trials. Then we took those characterized strains for screening in the greenhouse we established, and we used Leonard jars, we used pots and field trials to screen the best strains. And based on the Leonard jar, we have calculated the symbiotic efficiency for each strains. We calculated by directing the shoot biomass of the inoculated minus the shoot biomass of negative control divided by the positive control, which is supplied by the nitrogen. And we multiplied by 100 to give the symbiotic effectiveness. So those within the higher symbiotic effectiveness, we included and took for the field trials and pot trials. We did also for alfalfa, for common bean, for chickpea, for mango bean, and selected the best strains which are performing at the higher, which we call them high symbiotically effective strains. And then after defining those symbiotic effectiveness, we also developed carriers for inoculants. We used peat, we used charcoal, we used coal, we used compost and water hyacinths. We prepared locally, tested them, 5 different carrier materials were used. And we tested against 5 different legume hosts and different species. In all cases, the insect larvae pupa was the best inoculant carrier, followed by coal and others. And we used those 2 for our field trials. We also calculated the survival of the strains in the carrier, and we were able to define that they were able to grow 1 to 80 months up to 10 to the power of 9 log colony forming units for each cell. So we recommended farmers to use the inoculants if we preserve up to 8 months. Thereafter, the quantity reduces significantly. And then we also tried host by strain interaction studies, whereby the effects of host is inoculated. We included 5 different species, 8 different hosts, and we multiplied and applied in a factorial combination with 5 replications. And we did primary test in jars and then secondary test in pots. Based on our interaction, we were able to see that common bean is the best legume crop nodulated by most of the strains which are inoculated. The second one is cowpea. It was also able to be nodulated by a diversity group of species isolated from different legume species. Soybean is a relatively restricted host, and groundnut as well is relatively restricted host, including mung bean. Their nodulation also varies based on their association with the strains isolated from different legumes. We also try to calculate the shoot relative dry weights of the shoot biomass. And across all strains, we see there is a great interaction between strains and the Rhizobia strains in the field. So this tells us we can directly look for strains that correlate with a specific hosts. We try to see those really associating with soybean, with mung bean, with groundnut, and also with cowpea. So some strains, for example, CPW05 is best combining with soybean, and we also tried which one is where best. And with the common bean, these are one of the most important legumes diverging or associating with the legumes. And this enabled us to see and target for strains for location-specific legumes. Now we went for training after having the best inoculant from the effectiveness and also understanding their potential for different legumes. We went to train farmers. We reached 22 different locations and trained over 250 farmers. And we have also shown them in the field how we applied inoculants in a farmer's field. And then we brought farm inoculants from Nagasha Biotech Institute and BioSafe Inoculant Company. And this is our own inoculant that we have designed in the field, at Arbaminch University. And we took those to the farm and applied in the field. We prepared, showed the farmers how we can inoculate in the field. And we included our teams. So this is the sites where we tried our inoculants. The sites we see here is from Dembille site, which is one of the sites in Gamo zone. And we have also another site from Arguba, which have also different ecological views. And we grow there mango bean, groundnut, cowpea, common bean fields at Arguba site. We also did the field trial at Arbaminch Research Site, which is called Biological BB Diversity Research Center, where we grow mango bean, cowpea, common bean, groundnut, alfalfa in the field. And we analyzed, collected the data. Farmers visited to these fields, and also experiment was also conducted at Holte for chickpea and mango bean in this site. And farmers were also visited. We actually did the field trials at 7 locations, 2 in Arbaminch, 5 in different waredas, which are called districts. So from the field, we trapped it and uprooted the plant. We have seen extremely amazing nodulation with chickpea and groundnut, mango bean in fields. It's really very amazing to see huge nodulation effects. And we did a field trial, greenhouse trial visits along with farmers and selected officials to show what we have been doing in the region. And this is one of the fields that have been visited at Arbaminch main campus, which is one of our research sites. This is the field trials. One is from Arguba site, where we can see the mango bean which is extremely very productive compared to controlled trials. And this is one of the research sites at Arbaminch, and this is the mango bean with excellent biomass production and grain yield. We harvested along with the farmers. We showed and they compared the products that we have produced. We also checked the nodulation at each location, the biomass and the yield of the crops at those locations. And we try to see some of the preliminary analysis for chickpea, for example, and strain 73 was able to form excellent nodulation, nodule dry weight, and fixed nitrogen across locations. And this is really amazing. We have also seen the nitrogen derived from atmosphere. We measured it using KLDAL or wet lab analysis. And we also analyzed the symbiotic response, and it was really interesting that some of the yields were more than 10% when compared to the control ones. And this gives us a hope. Farmers were very happy with what we did, and they are asking for inoculants. And with this, we moved to calculate the yields performed. For example, for chickpea, the grain yield was between, when compared with the negative control, we start off the maximum yielding common bean strain. It was about 8.5 to 25% that yield varies across locations. And we are very happy that farmers are following our work, and we started developing relationships with other collaborators. We collaborated with Agricultural Research Center at Arbaminch, with Jinka University, with MBI, Menagasha Biotech Institute, which are producing inoculants. We supplied our inoculants for them. They can produce inoculants for us and share with us. And we also did field, exhibitions at different different symposia, workshops in Arbaminch and beyond. And this is one of the workshops that we have shown to the products. And on the table, you can see the inoculants that we produced at Arbaminch University and shared with the communities visited this company, this work. And it is really interesting. This is the flow that we have shown for the people and the improvement of the crops in the field. So this is also another show that we visited for the presentation of what we did in the region. So I really thank you for this, who are involved in this. These are the 3 PhD students working with me, and most of my colleagues are MSc students who are working with me. And I have also janitors who are working in the greenhouse and laboratories. So the team was actually excellent team, who are collaborating well with others. And we are working now with other teams in Ethiopia. And we are also planning to establish another project with friends to establish in Gofa zone, which will focus on mango bean in the near future. And I thank you very much for having me. And this was my final presentation. Thank you.
Thank you, Ashenafi. Thank you very much for this presentation and this broad overview of all the work that you have been doing, going from the farmer really to the lab, I would say, and bringing it back after to the farmer. This is fantastic, I would say. Very happy to see all this nice work. I'm sure there will be many questions, but what we will do now is, like, I propose the attendants to pose some questions in the chat, and we will answer them afterwards. After we also get the question from Sofie's talk. So without waiting, I would like Ashenafi to stop sharing your screen so that Sofie can then present the Belgian side of biostimulants. Thank you very much.
Okay, thank you.
Okay, hello, I will share now.
Yeah, the floor is yours.
I think I have to share this one. And now I go. You see the small one now, right?
Yeah, we see the wrong
Right.
one. Swap.
There we go. Yeah, it's okay now, huh?
Yes, it's perfect. Thank you.
Okay, good. Hello, everybody. So my name is Sofie Goormachtig. I'm going to tell you about an initiative that we started like 4 years ago about how to get inoculants for soybean when cultivated in our region. Now, just as a short introduction, so I'm from the VIB Ghent in Ghent, and Ghent is basically a village, not a village, a city, a city in Belgium. And if you would locate Belgium in Europe, then you come somewhere here. This is this little country here in the middle of all the bigger countries in Belgium. And Ghent is situated there, and we are a medieval city, and we are very famous for good food and good beer, such as there is beer, there is chocolate, there are French fries. We're actually famous for quite some, let's say, what I call the good life. Now, we also do science, and so in my lab, we're working on biofertilizers. So we basically look for soil microbes that can help plants to grow in, let's say, harsh conditions, which can be low nutrients, but also like drought. And today I'm going to talk about more the low nutrients, and this is the low nitrogen. Now, the project I'm going to talk about is basically situated within a societal and environmental challenge for Western Europe. And this is that agriculture and environmental sustainability should go much better hand in hand. What I'm talking about is the following, that in our regions, there is actually a massive application of chemical and organic fertilizers, and this has an environmental impact which is negative. And so Europe has already indicated that, well, we should reduce this because we basically put a burden on our environment. And so we have to, whether it's getting back or going forward to methods which are more sustainable for our life. So the European Green Deal, for instance, the farm to fork strategy, or vision text from the European community where this is expressed. Now, in the same region in Western Europe, we actually produce a lot of animal for meat, for human consumption. Humans, people also eat a lot of meat in our regions, and we also realize now that this is one of the causes that we have this environmental challenge here, and then we have to shift back, or we should shift back for more plant protein in human diet. So I really talk about here our regions that currently people just eat meat and it may way too much, and we have to reduce that again. So there is really a call for less animal protein in human diets. Now, together these two, less fertilizer and so more plant protein, this actually brings us back to the fact that legumes, and then we talk about peas and beans, and I think Ashenafi gave a good introduction there, have to get back more, has to get again a more important position in our agriculture. Currently in Western Europe, I think maize is really one of the main crops that is grown. The legumes, why legumes? Well, first of all, because the grain legumes, they make these grains which are full of plant protein, which is good for human consumption. Additionally, legumes, they have this capability to go in symbiosis with the Rhizobia bacteria. The Rhizobia bacteria fix atmospheric nitrogen into ammonia. So it's basically a natural way for the plants to get nitrogen fertilizer, and because of the structures which are formed on the roots, which is called the nodules, in which the Rhizobia bacteria fix nitrogen. So they take atmospheric nitrogen gas from the environment and turn it into ammonia, which can be used by the plants as nitrogen source. So this is the reason. So basically what I'm telling you now is that in Western Europe, the legumes should get a more important place again in agriculture. Now, one of the legumes which is at industrial scale a lot used is soybean. However, soybean is not so much produced in Western Europe. And if it's produced, it's actually more in the southern parts where it's warm. And then the problem now is that due to climate change, there's a lot of drought, basically also forcing us us to move some crops more to the north, and that's also for soybeans. So basically, again, here in Europe, there is an interest in increased production at northern latitudes in Europe. Okay, so growing soybean at northern latitudes in Europe, what does that include? Well, that means, of course, you have to deal with a couple of challenges, such as you have to develop a variety which can grow here. And that means it has to start growing in May, and it should be mature in September. So that means we need an early variety. And here in Flanders, for instance, but also elsewhere in Europe, there are some efforts to make examples there, new models, new varieties, and ILVO 3 is one of the ones we use here in our lab. This is developed by one of our colleagues from an agriculture institute. Now, at the same time, it's also important to basically look for a new inoculum, so a new Rhizobium, which can efficiently nodulate soybean when grown in our conditions. Northwest Europe means a lot of rain. It means colder climate in May. I think sometimes we really have still night freezing in the early May. So you basically have to look for a Rhizobium that can stand these conditions. And our colleagues from the ILVO Institute, they realized already that the inocula which were on the market suffer from cold, suffer from wet periods. So that's actually they're not so good when again it's raining in Belgium. At the same time, if we want to involve, or let's say alert people that they should reduce their meat consumption, we also should involve the stakeholders. And if we have the idea that they should eat more soybeans, then we also should inform them in time. So we looked for a project where we could also involve stakeholders such as the citizens as well as the farmers. Then in my lab, we also do molecular analysis for more efficient nodulation, but this I'm not going to talk about today. So in order to reach these two, we basically had an idea and we said, look, yeah, if we want to have a Rhizobium that can nodulate soybean very well when grown in our condition, we might actually look for it in our soils. It might be already present. However, we knew, since there was no soybean grown before and there's not much legumes grown actually which make symbiosis with its inoculum, we knew also that we probably were looking for a needle in the haystack. But nevertheless, based on what Becking and Beyering, two microbiologists, said already a long time ago, everything is everywhere but the environment selects, we thought we might find some good inocula somewhere at very low amounts. Now, since nodulation is repressed by nitrogen, so if you to nodulation, we knew also we should not go to our farmer fields because as I said already, our farmer fields have quite some nitrogen because of the application of the fertilizers. So we decided that it would be good to go into the gardens because in many gardens there are some spots, not everywhere, with very poor soils. Okay, and that's how we actually made the project. So by going to the gardens, we reached the citizens. That was number one. And we might have a chance to find rhizobia that we can then develop into a biofertilizer for our regions. So that's how we developed the Soy in 1000 Garden project. So we basically ask more than 1,000 citizens in Belgium to grow soybean in their garden. I will come back to that in a minute. And like this, we raised the public awareness about the power of soil microbes and legumes for a sustainable life. And at the same time, we also then got good trap, also what Ashenafi explained already before. We could look for rhizobia that can nodulate soybean in our gardens. We also were planning to get a lot of data so that we could develop models that might help us to predict rhizobial performance in various soils, so various soil conditions. So the first thing was, of course, and to go look for citizens. So we, by use of the minister and by a lot of publicity, we found very short, I think in like 1 month, we found more than 1,000 citizens who wanted to grow soybean in their gardens. So here you see that's actually half of Belgium, because if you go a little bit into the history of Belgium, you will see that Belgium is actually divided in two. There is this Flemish part and this Wallonie part, and there is a lot of politics in Belgium who tells that if you do science, you actually should do it or in the end one side or in the other side. And so we got Flemish money, and so we actually had to reach out to the Flemish citizens. You see, there is one citizen who has a garden in Wallonie, so he's a little bit our rebel in the whole collection, which is always very healthy, I think, in life. So altogether, we reached out to all the citizens. We asked the citizens to grow a square meter of soybean into their garden. So they got the seeds from us without any inoculum, so just the naked seeds. People were connected to a website where their seeds were indicated. We asked them to follow particular plants into their plot, and at regular basis they had to measure the plants, how many did develop, how many were germinating, how many had, what was the size, for instance, after 1 month, when did they see the first flowers, when did they get the first seed pods, and then at the end also the yield they got from their garden. So we basically connected to them, and so they collected a lot of growth parameters for us. So this was all done by the citizens. But then in July, we asked them, so after 4 months of growing, we asked 5 plants back to the lab. So all the citizens brought back 5 plants to the lab. So you can count already. I think we ended with like 950 citizens who brought back plants to the lab. And then we analyzed all those plants for nodules. And then I will come back to that in a minute. Now, in the nodules that we got, and this was for 106 gardens, we took those nodules, we cut them in 2 if they were big enough. If not, we actually had to choose. Half of those nodules went for microbiome analysis, and I will come back to it in a minute. And the other half went to isolation. So we basically started to isolate, and we got an entire collection. At the moment, we have 1,700 isolates from bacteria that were present in those structures. What do we find? We find all kinds of soil bacteria. However, for this study now, the Rhizobia are important because these are the symbionts of soybean, as well as the Rhizobia. These were actually our the usual suspects that we went on for further analysis. Okay, so we took these known genera, the Rhizobium and the Rhizobia, because we know that these are the ones that might form nodules on soybean plants. We took them and we brought them back to the lab. Again, very similar to what Ashenafi said, we did confirmation experiments in the lab, and we saw that some of these indeed made nice nodulation. And when it's red, it means that the nodules were red. And this is actually a sign for nitrogen fixation. So if nodules are red, this is because of accumulation of leghemoglobin in the nodules. And when the leghemoglobin dead, this is a correlation of the fact that in those nodules, nitrogen fixation is going on. So once we got some of those that were fixing in the lab, we basically went first on soils, later on we went to the fields, and I'm now really make a very quick shortcut on which Ashenafi actually extended for Ethiopia. But basically then 1 year later, we were now 1.5 years in the project, we did our third field trials, and when plants are fixing, they are dark green, so you can basically see on the color when something is going very well. And here you see this is the commercial inoculum here. Then you see the dark green. So this plot was actually fixing. And then you see here also the application of some local strains that seem to do very well. So this is then of course later then analyzed in detail. And this is just an example of one field where you see that the inoculum really increased. This is a commercial inoculum increasing the yield, but you see that our 2 local strains were actually doing very well in terms of increasing yield, as well as the protein content of the beans that were present on those plants. So basically, I can then tell you that via this trapping experiment, this is a very efficient way, and that's also what Ashenafi has told you, but we reached out to the farmers, to the people. So basically by soybean trapping in the gardens, by massive bacterial isolation, in 1.5 years, we actually had already a couple of lead strains that we now are currently using in agriculture fields, and that can be developed in an inoculant. Okay, so this was actually very fruitful and very effective. We were very happy about this. But at the same time, we also got a lot of data. And in the next part of my presentation, I would like to talk a little bit about the data that we got from this analysis. So in total, so the citizens, they collected a lot of data. So they measured plant height, their greenness, number of pods, number of when they were flowering. So we really got a lot of data from the citizens. And in the summertime, we also, when the plants came to the lab, we also measured the plant height. We also measured the greenness of the plants, which relates to the photosynthesis. We took pictures of the root systems, and we assessed nodulation. At the same time, when we brought the seeds to the people, we also collected soil samples from the fields or from the plots where the soybean would grow. So we also have a lot of soil chemical and physical parameters, such as soil type, different elements that are present, conductivity, pH, and so on. At the same time, we did microbiome analysis from the soil, so we have an idea about the bacterial diversity in those soils, as well as the fungal diversity. We also measured microbial activity by measuring phospholipid fatty acid methods, which gives you a little bit more insights not only in the presence, but also in the activity of certain microbial groups. So the people got a lot of analysis back from their own gardens, and they were very happy with it. So they also got some idea about the health of their garden. But we, of course, used those data for our scientific analysis. So basically, we got all the plants back in the lab. We assessed nodulation, and from 17 gardens, we got nodules. We could retrieve nodules that were red, indicative that were fixing. We also had 103 white nodules, and when you see a white nodule, this actually indicates that the nodule has been fixing, but it does not fix anymore, for one or another reason, or there is an incompatibility between the bacterial strain and the plant. That's the same for the brown nodules that we got. So brown also means an incompatibility. So we also got 100 brown nodules back. This actually indicates, I don't think they were ever fixing those, but that there is somewhere nodulation started, but it stopped for one or another reason because the interaction was not okay. And then we got everything like what we call a nodule-like structures. This might be little nodules, but it can as well be all kinds of, also, you know, just think about the nematode gall, because we really had to screen very fast, and the difference between a very small nodule and a nematode gall is not always so easy to detect. So here we call them nodule-like structures, but we are not always entirely sure whether they were really starting nodules or whether this is a kind of other interaction that took place. So we assessed the nodule phenotype. At the same time, we also did the isolations, as I told you already, but we also did the nodule microbiomes of at least these nodules that we analyzed. Okay, so what was then in total the outcome, the scientific outcome? I told you already about the application. So basically we have now 4 Rhizobium strains that really perform very well in our fields and will be developed in commercial inoculum. And we have a portfolio of 1700 bacteria that come from those nodules, and that might be developed in soybean biologicals, not really for nitrogen fixation, but rather for helping the nitrogen fixation to make it more efficient. What else did the project learn us? Well, we really reached out to the citizens. We made information sessions. We did reverse science cafes. These are cafes where the citizens come up with questions and they interact with the scientists to actually get answers to their questions. So this is very good for the citizens, but also for the scientists, because they actually then also know which questions to address in their science. We did a lot of questionnaires, and what we learned from all this is that our 1,000 citizens, that they were actually very good phenotypers. If we see, for instance, they measure the height of the plants at a certain point just before we got them in the lab. We also measured ourselves, and we actually got a very good correlation. So indicating that the citizens, by a lot of guidance, they actually were good phenotypers. We also learned that through the questionnaires that their objective knowledge about the nitrogen fixation and the soybean really increased a lot. Whether they're also more secure about their knowledge. This means that they talk to other people. They're much more secure about what they tell. So we learn them things. However, where they now eat less meat and more plant protein, these we had mixed results. And this has a lot to do with the fact that meat is very cheap in Western Europe because of subsidies that are actually given by the countries. And that in Corona times, actually, we got an increase in inflation, actually. And so that means that people were getting more poor. And then it's a more difficult situation to switch to plant protein. What now about all the science that we have done? And I will show you some data now. So I will first tell you more about the nodule microbiomes. So as I told you, so we did microbiome analysis of those nodules. We had 4 categories: the more necrotic ones, the fixing ones, the small ones, and then also the white nodules. And you can directly see, so we did a 16S microbiome analysis, which gives you insight in the microbial diversity of those nodules. And you can see that the microbial diversity in red fixing nodules is much lower at the alpha diversity compared to the other categories. Inside the white ones, you see there's still also some here with low diversity. The same is for the small ones and here, but you see here also that there are structures which are actually full of, or where there is a high diversity of different microbial species. This is somehow a confirmation of what we know, because when a nodule is fixing, there is actually one strain, or no, there is one strain who actually be at the majority there. It gets clonal, it will colonize all those cells of the plant, and this is actually the one that we detect, resulting in a lower microbial diversity that you see here. Now, then you can also ask the question, who is there? And I told you already, for soybean, we actually expect Rhizobium to be in those nodules. So this is only the red fixing nodules. And you indeed see that for some of those red nodules, there is actually one sequence that we get back in the microbiome analysis, which really corresponds to Rhizobium strain. And this is only for a few of those nodules, and I will come back to the other one there. So indeed, in some of those nodules, we got one main strain back, which corresponded to a Rhizobium strain. Now, you can then also ask the question whether also in the non-red classes we find Rhizobium back. And this is true for some of the white nodules, as well as for some of the unknown structures, as well as one of the necrotic ones. And these ones possibly correspond then to an interaction that somehow stopped because of maybe a possible incompatibility between the strain and the plant. Okay, getting back to the Rhizobium strains, which ones did we found? So here you see a phylogenetic analysis. We got 18 strains in total back. They belong to Diazophysians, Japonicum, Barani, and then one that we actually do not know yet what it is. It's possibly a new one. But what was very interesting was that if you look in those strains who had the genes for nitrogen fixation, this was only present in a couple of them. So you can directly see here that there are also some of the Brady strains that we found do not actually have the genes for nitrogen fixation, neither for entering into a symbiosis with a legume plant. So they are free living there, but they actually seem not to be very, let's say, evolved in doing a symbiosis with plants. Okay, now then I would like to come back to actually many of those red nodules, they had another sequence which was present at a very high abundance, relative abundance. And when we look to this sequence, we actually saw that this was tardivaha. Now, tardivaha is not known to do a symbiosis with legume plants, but if we did a phylogenetic analysis, and this was done already by colleagues some time ago, you actually see that tardivaha is closely related to the brother Rhizobium. And they seem to occupy red nodules. We also find them in white nodules. We also find them in some of the unknown structures as well, in a couple of these brownish nodules. And interestingly, there was one field study from the States where they also find tardivaha, however, at very low relative abundance. You see that here, this is the gray one that you see here. So then we isolated also tardivaha and we did some nodulation experiments. And when we looked at the data, we actually saw that none of them were actually ready to make red nodules on the plants in the lab. And when we sequenced the genomes, we also saw that they do not have the genes for nitrogen fixation. They neither have the genes for symbiosis, so the NOT factor genes that should be present. So basically, tardivahas, or at a majority inside nodules, the nodules were scored red, possibly means that there was fixation going on, but they do not have the genes for nitrogen fixation. So there it was a kind of a question we had. But if you then remove from our microbiome studies the main tardivaha and you look what's in the background, you actually always see that there is brother Rhizobium coming back. So this is basically your relative abundance here after we abstracted the tardivaha. So you really look into the minority of the sequences, and if you look into that, there is always a brother Rhizobium somehow into the background sitting there. Opposite, if you look in the nodules where brother Rhizobium was most abundant and you take brother Rhizobium away, you always see that there is some tardivaha there in the background. So it really looks like these two are playing the game for nodulating soybean in our conditions. So in total, I can tell you that only in few of the gardens, and this is only 17 from the 1,000, it's not a lot, we found nodules back on the soybean plants. So you can really say that in Flanders, our soils, they're really blind for soybean. They're really not adapted for soybean, and there is no natural nodulation going on. So we've got only few strains back, but few is enough for applications, while we also found some non-nodulating bradies. So there's really no co-evolution or co-adaptation between the plant and the soil microbiome. Now, there is another question that I think we see here. Fixing nodules have one main strain. That means somehow this strain is able to occupy the niche and to assure its growth niche. How this works is still something I think we should figure out. And we also see when nodules get non-functional, there's a high diversity of microbes observed. Or these opportunistic ones, we at the moment do not know and should actually look at that in more detail. Now, tardivaha next to the non-fixing bradies is also a very interesting one. It's present at high relative abundance in nodules that look red. What does that mean? Is it a helping strain? Does it help the brady strains which are at lower abundance? Is it what we call a cheater strain? Because evolutionary wise, if you think about maybe about the advantage of a rhizobium inside a nodule, maybe tardivaha is a super good cheater strain. So it basically cheats the soybean. It lets the soybean think that it's going to do something good for the soybean, but at the end of the day, it's actually a cheater. And it gets all the sugar from the plants, but it does not give anything back. And so this is what we call the cheaters. So basically, squat the world. And this might be actually something that tardivaha is doing. And this is something that we're going to figure out now in the future. So altogether, I think we have seen here already that 17 from the 1,000 soils, this is really not a lot. So it really shows that our soil microbiome is not adapted to soybean. Now, in order to demonstrate that further, we basically said like, well, let's take 100 soils and we grow Pisum P, which is a well-known legume for Flanders. And we again grow soybean. And then you can see here, we took 114 soils. And just to show you here again, so only in 23% of the soils, soybean got nodules, and only I think in one of those, we actually got red nodules. Really showing again that our soils are not adapted for soybean. If you take the same with pea, 93% of those soils get nodules, and the majority were actually red, indicating for nitrogen fixation. So it shows again here, well, if you move a plant, you also really have to think that the soil microbiome has to get adapted. And then if you look into those microbiomes, the pea microbiomes really illustrate a typical healthy fixing nodule with one main strain here, which was a Rhizobium strain. And if you look into the microbiome of the nodules we got on soybean, you see a high diversity. So again here, this is indicative for a non-functional symbiosis, and actually many strains that seem to occupy those nodules. Again, here, some of you might think, yeah, maybe there is just simply no Rhizobium present. Well, if you look into the relative abundance of Rhizobium, we indeed only, they dropped off the limit of the microbiome analysis in over about half of the soils, but it's not because it was present that you got nodules. So they're there, but I showed you already, yes, some do not even have the fixed genes, so this is the case. The relative abundance of Rhizobium is a bit higher, but it's still very low. So you see here we are 0.1% of the total. And this is, for instance, a bacillus here, which goes then to 1% of the total. So altogether, I think I already addressed these two parts here of the science we have done. Now, what about soil factors influencing nodulation? So then here our system biologists of the lab come in, and so they try to correlate soil parameters with the presence or absence of nodules. Now, the different models that I have been using are, well, they are significant, but not highly significant. That means that there are still many parameters we don't know what actually drives the nodulation. But I can tell you that, for instance, low nitrate was inversely correlated with nodulation. And this gives me confidence of the data because this is well known. And it's very interesting to see that in Flanders, well, if you go from left, this is France, this is Holland, you get actually a lower level of nitrogen into the soils, and this gave you a higher chance of finding nodules. Also, this is actually, for me, this was an assurance that our data set is really robust. But you see that we get other correlations as well, such as low phosphate, low potassium, also the conductivity, the salt, and some of the pH here. But these are the factors that we now have to further, so these are negative correlations that we have to further address. What was also very nice was that we get a positive correlation with a high diversity of microbes in the soils, but not especially Rhizobia. So the higher diverse the soil is, the higher chance that you get for nodulation. So what is now the future of our data? Well, the future is that this is just an example of two field trials. Just looked at the protein yield in 1 year, and then in the other year, you see there is a high diversity here, which is really depending on the environment. And then I talk about the weather. And this is something we at the moment really have not much clue about what's going on. So this is something to address in the future. So what we will do, we just have a new project where we're going to reach out to the farmers. So pretty much what Ashenafi has been doing in his project. We're going to look much more in the cause of the variation. So we're going to, let's say, collect much more data also on weather conditions and to see what's going on there. Now, what's the role of the non-fixing Rhizobium strains and also the tardivaga as a helper or a cheater? This might also be a reason why we have so much variability in the field. We're also going to reach out to the citizens, so we're doing more information campaigns and more actions. And of course, we also want to reach out with our expertise and experience to the world. And then we have now an interaction with Ashenafi, and we hope we can merge all our expertise together in order to make our world more sustainable. And I would like to end with this. Have a look to the movie.
Not really going through, Sofie.
You see it? Ah, okay. Then I go because you don't hear it. Okay.
Yes.
Then I will just move. But what happens here is that my people made a movie about biologicals in agriculture. They won 5,000 euro. They won 5,000 euro from a nature campaign. And like this, you know, they made bigger impact for biologicals in agriculture. And so that I just wanted to show you that it's not only the citizens we reach out to and the farmers, but also my people in the lab. They get all educated to try to make impact. And this is what they did recently. And I'm very proud of my team. And like this, I would like to finish my talk here. So this is a big collaboration with many people from the lab, not only from my lab, but also from the universities in Leuven and Ghent and ILVO. And so I'm open for questions.
Thank you, Sofie. Thank you both, Ashenafi and Sofie, for a very nice example of citizen science, inclusive research where people and stakeholders are involved, and both stories ending with potential products actually for the farms. There were several questions for Ashenafi. So while people are also writing down questions in the chat for Sofie, let me start already with some question for Ashenafi. Let me take those. Yes. So from Val, we got several questions. Actually, he was saying, "Hi, Ashenafi, very interesting talk. Did you do some soil analysis? I think you did, but you didn't much mention more about it. So that's the first thing. Did you do some soil analysis? Second is, do you plan some microbiome characterization? It's talking about meta barcoding to follow your inoculants. So maybe we start with those two. So did you do some soil analysis? Ashenafi, you here?
Yeah. Yes, I do. Okay, thank you very much for the questions. Yes, we did soil analysis for different locations, but for the sake of the time, I didn't include in the, we also did some correlation analysis, and pH, altitude are really affecting the variations we saw. Mm-hmm. And with the barcoding, we didn't do because the facilities we have didn't allow us, and that's why we are looking for collaborations to do in the future.
Exactly. So you said, interestingly, in groundnut, you said it's less permissive regarding strains, and he says that he observes different things in Senegal. And the question that comes with it is, do you follow nitrogen fixation in the field, like chlorophyll measurements or things like this, plant parameters?
Yeah, we did not chlorophyll, but we measured the nitrogen fixing. We collected the plant biomass from the field and analyzed the shoot nitrogen content using the Kaldal wet method analysis. With the low, because we compared with other legume species, such as common bean and cowpea, which are nodulated by a diversity of legumes. Strain is coming from different host sources. So it's in terms of promiscuity, common bean and cowpea is more promiscuous, nodulated by different Rhizobium species, but soybean in my case was restricted. It could be probably because in southern Ethiopia, soybean is less adopted in the region. And that's why we have, as Sofia mentioned, in Europe, you don't have much soybean strains in this field, and we didn't have also this that much. And we collected where the soybean has been introduced previously in different locations, like in Western Ethiopia, in some parts of southern Ethiopia.
So you're saying that you have been collecting in places where where legumes were already cultivated, but were those legumes already inoculated, or is it like just pure cultivation of legumes but without inoculation?
For our case, it's introduction of legumes where they are growing, but inoculation didn't happen in our region because the area didn't receive any inoculant, except we found some inoculants in hands of the extension experts, but they didn't introduce to the farmers.
Okay, so from Lucy Mushiri from Kenya, we do have two questions also. What are the possible reasons that the common bean has more Rhizobium strains than than other legumes? And did you check whether the Rhizobium was including host defense against the biotic factors in legume production?
Okay, thank you. Common bean is really well adopted in Ethiopia. It's in 1970s that it was introduced to Eastern Africa. Sometimes the people mention that it could be in 14th century that common bean is introduced, and it is much more adapted to the region, and it's one of the common legume crop growing vastly across the region. And that's why there is a huge adaptation of the legume with the strain or rhizobia in the soil. We didn't actually test any, we tried to analyze some of the plant controlling molecules which can be produced by the plant when they have association with legumes, or we can test the metabolites from the rhizobia in the lab, and we check it for indole acetic acid production, for siderophore effects in the lab. And these are some of the reasons that can enhance. We can also analyze some other chemicals, but we didn't correlate to the abundance or to the distribution of common bean rhizobium.
Okay, thank you. Let me move a bit to Belgium with a question for Sofie. Sofie, Val was asking, how long for microbiome adaptation, to your opinion?
Yeah. On my opinion, I don't know, but we will. We have now a new project where we will measure this. So the good thing of this is that scientifically we can follow this now, because we are starting with this blind soils and we can now actually follow it year by year. But I can't give you an answer yet. So we will. This is really one of the topics of our new project now.
Okay. From Lucy in Kenya, who is a nematologist, actually, she is asking, how did you differentiate the nodules from root nematode gall? How did you make that different?
So that's a very good question, because this was really one of the things we did, one of the aspects when we were screening that we really looked at. The nematode galls, we usually saw many on one root system. You know, we saw like when we saw many on a root system and it really looked like a nematode gall, we actually put a plant aside, because usually when we were screening and looking for nodules, we only got like 1 or 2 more nodules per root system in this trapping experiment. So that was one of the reasons why, one of the ways we did. But that was all, because we did not have time. You know, we had to do 5 times 1,000 plants, 5,000 plants in like 3 or 4 weeks. So we did not have time to look more in detail.
Okay, thank you. I think I have a question from John, and it may be addressed actually to both of you. He's asking about the strain efficiency. How long does that last? I think Ashenafi has been mentioning something about this. Maybe he can start.
Yes, we try to see the survival of the strains in a carrier that we have been using. And we tested in the lab every 2 months for the survival, and we calculate, we estimated the colony forming units from each strains. And for our strains which have been used with the coal as a carrying material, we estimated 10 to the power of 9 cells up to the 8 months, but thereafter they gradually dropped. They dropped the number of the unit, colony forming units dropped significantly. So we thought it is good to use inoculants up to 8 months.
Okay, but that is independent on the storing conditions. That's really like, because yes, I suppose that is an issue, right?
Yes, we use 2 conditions. One is open space in the dry room at room temperature. The other is we use refrigerator to compare the data.
Sofie, how do we do that in Belgium? And do we have a very different approach and lasting of our strains, or how does that go?
Well, we still have to measure it. So we did not do these experiments yet, so I cannot tell that. When it comes to storage, so also here, if you want to coat your seeds, you need to have like a 2 or 3 month period where they survive. Now, this is something, because in Belgium, you know, the farmers get it from the companies, and the storage problem is more a company problem than my problem. In short, it's a bit a strange way to say, but so for our field trials, we use a very basic coating which works, but actually for application, I know the companies have their secrets, which I don't know how that works, to make them to have a longer shelf life, right? Yeah.
Okay. I went through quite some questions, but I mean, people, I would encourage you to raise your hand if you would have a question and to build on the conversation. Please do. We still have some time to continue. Just raise your hand and we will give you the word. Meanwhile, Sophie, yeah, I had a question. Is it true that local isolates are not effective in NF, so nitrogen fixation, I suppose? Some research has also shown that commercial inoculants have failed, especially when they are not tolerant and when their genetic stability is poor. This makes native strains to be more effective. We need to look into that. Any feedback on the difference between native strains and what is then called more commercial strains, I would say?
Yeah, so at least in Flanders, we know that the commercial strains really suffered when in May the time was very wet and cold. So this is true. We also have, I think, Ilvo, our colleagues have these studies done. That's why we went for endogenous strains. But you also, I know from colleagues, for instance, in France, that the commercial inocula might actually stay in the soil and then get inefficient again. So, and I think we should never forget the biology of the symbiosis. I think the cheater hypothesis might from the perspective of the Rhizobia actually be not a bad idea. You go into a plant, you create a niche, you get the sugar for the plant, but you don't do anything back. So that might be actually a thing that evolution goes to. Of course, plants will react, and that's then how you get this plant-microbe interaction. So definitely also local strains might not necessarily be the most effective one. So I think it's something to keep in mind, to keep on checking. And I think depending on which field with which history, the problems will be different. Like for soybean, we have to add strains, but I think for some legumes, you might actually have to add a strain which is very efficient in infection. So it actually overrules the local ones, and it's then also very effective in nitrogen fixation.
So,
I don't know if this helps.
Yeah, I suppose it does. I would follow up on that. And do we have like an idea of the time, the lifespan of? I mean, if you say it's in the soil and at one point it might get inefficient. I mean, it's kind of an evolutionary approach of your soil, I would say, or even pollution, if you want to say in some way. Do you have an idea of how long that would take so that?
No, I don't have. And it's actually not only, how to say, there's also a lot of horizontal gene transfer between strains, right?
Okay, yeah, that complicates the story.
The soil is not it's a microbial pool.
Yes. Val, you have raised your hand. You still have a question for us?
Yes, I have maybe more comment. I will say I'm really thanks to both presentator today because the talk were very interesting. So presently I'm Valérie from IRD in France, but I'm based in Senegal at the moment.
Hi,
and
Valérie.
Hi, Sophie. How do you do? I'm very interested by the cheater things because that's what we have here. I'm working on groundnut. And we isolated different strains here, and it appears that some of them are cheating and not efficient, but doing this, what you call so-called nodules. And we are working at the moment on the microbiome story, but also on the genetic of the plants, because it appears also that according to the genotypes you are using, the microbiome that is captured by the plant is not the same, which can also it's also an interesting piece to look for, because can be also some the problems of adaptation or recognition between the plant and the microbiome. But thanks again. It's very excellent
Yeah, it's really interesting,
information.
Valerie, what you say with the connection with the genotype. What we see here also in the lab is that soybean is super, how do you say, promiscuous compared to many other legumes we have been working on.
Yeah. It's the
Which
same
might
for,
also be important
yeah,
in terms of cheating.
the same for groundnut at Lucid Senegal. So that's why I was very interested about the result from our Ethiopian colleagues, because it doesn't exactly see the same. And I think it can be linked to the difference so between the plant genotypes maybe. And of course, what is in the soil also. Thanks again.
And maybe a question for the two. If we look at those populations of Rhizobia and so on, if we think of climate change, because this is what is happening, Sofie has been mentioning it, we are changing from one area to another to cultivate the soybean. But what if we want to cultivate, let's say, we still want to cultivate in south of France where it's getting dry. We still want to cultivate the soybean. So what do we do there? Because the Rhizobium is not anymore efficient, probably because of this climate change. How do we approach this? Can we find other ways, or do we need to import, let's say, the Rhizobia that comes with it? How is that? I mean, in Ethiopia, that is a very important case also, because you have very high climatic differences between the regions, right? So how would you approach that? Maybe Ashenafi first.
Yeah, okay, thank you. Yeah, truly the climate change is affecting both local communities, and the target is to use a legume inoculant, and there is one thing we can make sure that if we introduce strains into the soil, there is a horizontal gene transfer between strains, and the nodulation genes are capable of, because they are on the mobile elements of the plasmid, they can transfer. For broad Rhizobium case, they are on the chromosomes, and there is a less efficiency of transferring of the genes. So there is still a possibility of transferring the genes into the naturally adopted strains. And growing crop is consistently in the field. I did another experiment with a colleague from Hawassa University. We tried isolation of strains which were grown consistently for a longer time in the soil. And we found local strains which originated from tree legume were capable of nodulating chickpea in the field. And it was for the first time that we reported the tree legume, which is within the Rhizobium species belonging to tree legume, was able to nodulate chickpea. So I'm mentioning that it's possible if we grow consistently legumes in the soil and inoculate ones, they can be adopted in the soil and gradually develop to build up in the soil. So it's possible to introduce in the locations. One of our future targets is we are trying to introduce a massive production of common bean and groundnut, soybean in the lowlands of Ethiopia, because they are also drought-tolerant crops, and they can also associate with different rhizobia in the soil. So this can improve our response to climate change.
And I think we should not forget, for instance, the mycorrhiza, Ashenafi as well.
Yes.
So together with the Rhizobia inoculum, the mycorrhiza can then also help for drought resilience, right?
Yeah.
For instance, or the streptomyces, or yeah.
Yeah, so as Sofie mentioned, we are also planning to have a combination or a co-inoculation experiments with Rhizobia and AMF, which extends the nutrient absorption capacity of the soil, is capable of extending the root system of the legumes as well as other crops in the soil. So this can also enhance the survival under the drought conditions.
And continuing on this, if I may ask, from a very naive point of view, because I know nothing about mycorrhiza and so on. For example, in Ethiopia, you have like very drought-resistant grass pea, so Lathyrus, which is very, very drought-resistant. Would there be potential to get the Rhizobia from these and apply them to other crops that are more common? and to use them to other crops that are not common, or is that not compatible at all? I mean, maybe I ask just a dumb question and you will apologize me for that, but how can we take from other legumes some rhizobium to apply to other crops?
Yeah, we can do this if we only test the host range test for that specific group, and we can find related hosts and evaluate the strains that are capable of nodulating those legume species. Otherwise, we can go for gene transfer mechanisms to look how we can insert some nodulation and symbiotic fixing genes into the strains and apply into the field.
Yeah.
Good, thank you very much. Unless there is any additional question or comments within the audience, then please raise your hand now. And I think there is none. So if not, I would like to close this seminar by thank you both speakers and participants for the interactions and a very nice story around biostimulants in Africa and in Flanders. Thank you very much.
Thank you very much.
Welcome. Bye-bye. Thank you.
Thank you.
Bye-bye.
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 Bye.